Hi students, welcome to HSC Biology and Module 5 on Heredity. This is video number 23 and we're going to be looking at incomplete dominance. As we've looked at already, there's been a bit of a look at the Mendelian ratios and how Mendel explained the inheritance of different factors when he was looking at experiments involving pea plants. Since then, we've looked at a number of different types of inheritance that deviate from those Mendelian ratios. And this is another example. This one involves incomplete dominance. So this is basically the same sort of theory that you used for codominance. So we're going to find the same sort of things happening in this particular video as we did in the previous one when we looked at codominance. The difference again is the interpretation of the heterozygous condition. So what we're going to be doing is seeing if we can solve some problems using Punnett squares in order to look at specifically incomplete dominance. So I guess the most important thing for us to do when we're looking at incomplete dominance is to contrast it with codominance. So you remember with codominance when we had a choice of red and white, and we said they both have to be homozygous if they're going to be red or white. When they were heterozygous, and that is one example of each, then what we had was a situation where coat color, for example, displayed both red and white hairs. So both of the two alleles were equally expressed. The problem this time is that when we have the heterozygous condition, so when we have a RW or a big R, little r situation, what's happening now is the dominance is incomplete. So we get something in the middle, some sort of intermediate. So snapdragons are a good example because snapdragons produce pink flowers. So pink is not red and it's not white, and it doesn't have like striped patterns or spots or anything like that of red and white. It's a complete blend of these colors, and so we get pink. We get something in the middle. So then if we're going to, I guess, define this, then in incomplete dominance, neither of the alleles dominate. So when the heterozygous condition occurs, and of course these sorts of variations are only relevant to the heterozygous condition, and when it occurs we get this intermediate phenotype. So something that is between the two allele expressions, but something that doesn't actually represent the expression of both. So that's the key difference between incomplete dominance and codominance. And I mentioned the fact that snapdragons are another kind of red-white combination and a good one to contrast with our roan cattle, because in the roan cattle both the red and the white colours appeared in the coat. In this case, neither the red or the white appear. We get something in the middle, so we get these beautiful pink flowers instead. Now, if you're following through, and I've just done this exactly the same way as I set out the problem for codominance, so you can see we've got to start with our pure breeds. Our pure breeds are only going to give us one option for the gametes, because remember the gametes are about splitting that information in half, and we do it randomly, so we get any possible combinations. But if there's only W's, you can only get W. If there are only R's, you can only get an R. And so therefore in the F1 generation, they're all hybrid, they're all heterozygous. That means in this case, if we're talking about snapdragons, then the color will be pink. So pink corresponds to the heterozygous condition. When we do the F2 generation, again, because we've got a genotype which is heterozygous, that means either of these two alleles could go into the gametes for both the male and the female, and we need to take account of that all possible combinations. And so that means that we have our Punnett square again, which has a 2 by 2. So we've got our male or our females on either edge, and we know that both of these different alleles are present, and so therefore the combinations that we have again, as we did or as we looked at for codominance, was a 1 to 2 to 1 ratio, which is our Mendelian ratio. And again, the problem is that the interpretation of genotype to phenotype, that's the only thing that changes. So we've got one white, we've got one red, and then on the diagonal here, we have our two hybrid conditions, which in this case would be pink. So the only difference that we're looking at between codominance and incomplete dominance and complete dominance is the interpretation of that heterozygous genotype. Practice a few of these. There's probably more examples of incomplete dominance than there are of codominance, but it's all about being able to interpret that genotypic ratio through to phenotypic ratio for these types of genetic problems. Thanks for watching.